So welcome back from the break. Let's take a look at the concept check question. Where in the life cycle do we see reflected the 5 derived adaptations that are common to all seed plants? Well, let's go back to the life cycle. It's the previous slide and let's take a look. So remember what those 5 derived features are. A reduced gametophyte. So where do we see the reduced gametophyte? Well, the gametophyte is going to be located within the microsporangium initially, and is also going to be located within the microspore, which is going to give rise to the female gametophyte. So where do we look for the reduced gametophyte in the microsporangium? Well, that is within the pollen grain. The pollen grain itself is the microgametophyte. So we would expect to see the microgametophyte in the microsporangium. We expect to see the megagametophyte, which is the female gametophyte, within the ovule itself. So there's where we see the evidence of the reduced gametophyte. We also should see the evidence of heterospory, the heterosporous condition, meaning microspores and megaspores. So where do we see the microspores? Well, the microspores are within the microsporangium, within the staminate cone. And so we can see the microsporangium and the microsporocytes, which via mitosis give rise to the microspores, which develop into the pollen grains. So that's where we see that. The megaspore is found within the ovule, product of the megasporocyte within the megasporangium. Again, via mitosis, here is the megaspore within the ovule. So now we've seen the appearance of the heterosporous condition and the two spores, the megaspore and the microspore. That then leads us to the origin of the ovule and the pollen grain. So the ovule is what gives rise to the seed. The pollen grain, with its means of distribution in the absence of water, in this case, transmission via wind, are real advantages. Remember, it's the pollen that gives us the ability to reproduce in the absence of water, and it's the protection of the ovule within the body of the plant and allowing the embryo to develop within the ovule that is recognized as a derived trait that leads to the success of seeded plants. And finally, there's the seed itself developing from the ovule. So we see the seed appearing over here, drying out of the integuments to give rise to the seed coat. So this is our seed developing here. Containing or packaging the embryo, and this is what is, with the food resources, this is what gets released into the environment, again, considered to be an advantage for seeded plants, and one of the reasons why they've been so successful as colonizers of dry land. So we see all 5 of those derived adaptations reflected here in the conifer life cycle, or the pine life cycle. So now let's take a look at angiosperms, the largest group of, or the largest clade within the plant kingdom. So the term angiosperm comes from the Greek word for flower, and it is the flowers that are the distinctive feature that most people associate with angiosperms. But in fact, there's a lot more associated with angiosperms than just flowers, because if we think in terms of fruit, what we have to recognize is that you can't have a fruit without a flower, because a fruit is the mature form of the ovary, which is the protective female structure located within the flower that protects the ovules. And this is why the term angiosperm refers to, or often is translated as covered. And I got it wrong. Angiosperm does not does not come, or the angiosperm does not come from the Greek word for flower. The Greek word for flower is anthos. So, so the phylum name I think it should be one L, shouldn't it? Not two. Let's clean that up. So the phylum name is Anthophyta. But angiosperm means covered seed. And that's because the ovules now are located within a protective structure called the ovary, which is part of the carpel, which is the female part of the flower. So it all hangs together. So I've just covered that. So let's move on. So when we look at the parts of a flower, what we need to remember is that all parts of a flower are derived from sporophylls. And in fact, a flower is produced by a transition and the same is true also in the case of the pine cones and the strobili that we saw in the gymnosperms. There's this transition from what is known as vegetative growth, which produces stems with leaves and so forth, to floral growth, which produces the cones of the gymnosperms and produces the flowers of the angiosperms. And it's all about changes in gene expression that leads to the modification of the sporophylls to these various structures. Now, some of these structures do look like leaves. So the sepals look very leaf-like, the leaf-like structures that cover the flower bud. The petals look very leaf-like, and in fact, the petals contain chromoplasts. These are plastids that contain flower pigments. So these belong to the same family of organelles that chloroplasts belong to. But rather than being specialized for photosynthesis, they contain other pigments that give the flowers their characteristic colors. It may look like the carpel and the stamens are not related to sporophylls, but in fact, we know from fossil evidence that the carpel itself is probably derived from a bundle of leaves that folded together to protect the ovules, and over time, they evolved into this more formal structure that we recognize as the carpel. We do also notice, as part of the evolution of flowering plants, that the covered area, what would ultimately give rise to the structure that we call the ovary today, started as either uniformly distributed along the length of the carpel or may have actually developed more towards the upper end of the carpel, but the evolutionary trend as identified through the fossil record is that the ovule descended to the base of the carpel. And so that leaves at the top of the carpel a landing zone for pollen known as the stigma. The style connects these two together. The pollen in this case, and I mentioned near the beginning of this presentation that the germination of the pollen would be a little bit different in angiosperms. The pollen never actually enters in to the ovary, nor does it enter into the micropyle at the base of the ovule. Instead, the pollen remains seated on the stigma and germinates there. And the pollen tube, which still is produced, grows down inside the style around the edge, the fleshy edge of the ovary, and then penetrates into the ovary and grows in through the micropyle at the base of the ovule to eventually discharge not one sperm nucleus, but two sperm nuclei into the ovule. Because within angiosperms, it's not a single fertilization event, it's a double fertilization event. We'll talk more about that when we look at the life cycle. So when we start looking for the gametophytes and we start looking for the sporangia, we look again within the ovule for the megasporangium. We look for the microsporangium within the anther. So the microsporangia with the anther. As soon as we say that, of course, we're also tagging the stamens and the carpel as sporophylls, because remember, sporophylls are the leaves that are modified for reproduction by the presence of the sporangia. So the moment we say that the megasporangia are within the ovules, within the carpel, within the ovary as part of the carpel, and that microsporangia within the anthers as part of the stamen, we're also saying by implication that the stamen is a highly modified sporophyll and the carpel is a highly modified sporophyll or cluster of sporophylls, which is what we really think is the case as far as the carpel is concerned. When we look at the arrangement of petals in the flower, the flowers can either have a radial symmetry, so if we draw lines from the center of the flower outwards, you can see the radial symmetry reflected here. Or you can see a bilateral symmetry, which if you draw the axis down the center, indicates the bilateral symmetry. We also notice that in some situations, flowers can be unisex. So here are some holly flowers, and these flowers are growing on the same plant. So you have male flowers and female flowers growing on the same plant. These flowers lack a carpel in the center. They only have functional stamens. So these would be male. And the female flowers have stamens, but they are non-functional, but they do have carpels. So these would be female flowers. There are some species of flowering plants where there are two separate genders. So there are female plants and male plants. In between my undergraduate degree and beginning my PhD, I spent some time as a volunteer teacher in the South Pacific. And the school that I was teaching at had a farm, and we used to grow produce to ship to New Zealand to help earn some income for the school. And we used to grow papayas or pawpaws. And in papayas, you do have male trees and you have female trees. So you would think perhaps, well, maybe if you just grow the female trees, you can maximize your production because it's the female trees that are going to produce the fruit. But you also have to remember that the fruit's not going to develop until the eggs within the ovules are fertilized to develop into seeds. So you do actually need male trees, you just don't need a lot of them. And so in the average pawpaw plantation, like the one that I was involved with, we would have about one male tree for every 10 female trees to provide the pollen necessary to fertilize the flowers so that the ovules would develop into seeds and would trigger the development of the fruit, because the development of the fruit parallels the development of the seeds and the growing embryonic plant inside the seeds. And this is the reason for the double fertilization, so that the two steps occur in parallel, so that when the little embryonic plant reaches its mature point, the packaging of nutritional material into the seed, the endosperm, also reaches mature point, and the fruit can then mature to trigger whatever process is going to lead to the distribution of the seeds. So we want all of these processes to occur in parallel and in a particular sequence, but none of it begins unless the eggs are fertilized by the pollen. So you do need male plants around, even if your agricultural product is coming from the female plants. So speaking of fruits, fruits are the mature ovaries. So when you eat a fruit, you're actually eating a mature ovary. Think about that next time you eat an apple or an orange or a tomato, because a tomato is actually a fruit, or any other fruit that you may enjoy eating. You're actually doing what the plant intended you to do, consume the fruit. So why consume the fruit? Because in the best of all worlds, you would aid in the distribution of seeds, because you would consume the seeds as well. Maybe you might destroy some of the seeds, but some of the seeds would remain intact and pass through your digestive tract and be excreted and likely be delivered to a new location, exactly what the plant is looking for. You're actually aiding in the process of seed distribution. This is the reason why fruits exist. They're not there to act as a nutritional food source for animals. They're there to attract animals by offering them an incentive to consume the seed-bearing part of the plant, the ovary, and therefore contribute and aid in the distribution of seeds. So it's all about the plant manipulating their animal collaborators so that they will consume the seed-bearing tissue and therefore carry the seeds with them. So here you can see some fruits. You should be familiar with these from doing the lab last week. There's the tomato. So you can have fleshy fruit or you can have dry fruit. Some dry fruit remain intact, like nuts. Other dry fruits split open to release the seeds, and you can see the milkweed doing that here. The actual seed, or the actual, I guess the seed, as far as the nectarine is concerned, is this, the pit inside the fruit. This is the ovary wall that has matured and become fleshy, and that is what you consume. Here is the ovary wall out here in the tomato, and then you have the seeds within the ovary. So these are the mature ovules, and they're broken up into compartments. In the grapefruit, again, you're dealing with the fleshy part of the ovary, and the outer layer of the ovary develops into the skin. This is the inner portion of the ovary that becomes the fleshy fruit. The ovules, the seeds, are actually located here in the center. So depending on the type of seeds and the type of fruit that are involved, there are many different ways of distributing seeds. All are adaptations to guarantee as wide a distribution of seeds as possible. So some are wind-borne. So if you find a dandelion and you blow on the dandelion, on that little cotton ball that develops from the dandelion, and all those little fluffy things that get released into the wind, you've done exactly what the dandelion wanted, because though at that little brown knob on the end of that floating, that fluffy part of the dandelion, that's the actual seed, and that fluffy part is an extension of the integuments and the seed coat. And you've now just released that seed into the wind to carry the dandelion seed as far as possible to increase the distribution of the dandelions. You've done exactly what the dandelion wanted you to do. We've talked about animals consuming fruit, and here you have some berries, some blackberries and raspberries and so forth. Each fruit is actually one of these. Let me see if I can do this. I'm not sure whether I can do this. So one of these little black swellings is actually the fruit. And within that fruit is the seed. So this is a multi-part fruit. Each of the little black parts is actually a single fruit, and you've got multiple fruits here, each containing a single seed. And then I love the look on this dog. Being a dog owner and someone who's been around dogs all my life, I can just imagine this dog going, what on earth have I done to deserve this? Because this is an enlargement of this seed, and you can see the little hooks on the ends of these barbs. That's what's caught in the fur of the dog and has meant that this seed can now be carried from one location to another. If this looks somewhat familiar, or maybe it doesn't, this actually has a significance. And I'm not sure whether you would be familiar with this, but you've almost certainly used an industrial product that was developed by understanding the significance of these little hooks. Let's see if I can do this. So Velcro actually has a very old origin, or at least the idea of Velcro has a very old origin. It was first thought up by a Swiss chemist who was looking for a new and novel product that he could patent for his company. He went out walking with his dog, and his dog and he suffered this problem with these barbed seeds attaching to the dog's coat and to the man's pants as he walked through the field. And he got to wondering why these seeds were constantly attaching themselves to his dog and to himself. So he put them under the microscope and saw all of these little barbs. And he started wondering if it would be possible to replicate these little barbs and some sort of loop structure that he saw in the surface of the material of his pants that would allow the barbs to hook into it. And so he wrote up a description of a product that used this hook and loop structure. He didn't call it Velcro. Velcro would be the name given to it in its modern form, but he first wrote up this patent in the 1930s. However, materials did not exist at the time that would allow for the development of the product as an industrial product. So the patent lay dormant for many years until NASA came looking for some way of making a reusable fastener of some description that could be easily opened and closed again in weightless conditions. And this is when Velcro was originally developed. Yes, it is a product of the space program, because NASA was the launch customer for Velcro because of their requirements for an easy to use fastener that would be easy to work in weightless situations. But the idea has a very old origin going back to the late 1930s. Kind of cool. This is not the only time we'll see plants and humans having some form of interaction. More to coming towards the end of this. So let's take a look at the angiosperm life cycle in this diagram. So this cycle usually plays out over the course of the growing season for a plant. So for an annual, that could be as long as 12 months, but for annuals that grow in, say, Alaska, the growing season could only be 3 months long. So this whole life cycle could play out in 3 months. Again, we'll start with the sporophyte. So the sporophyte and the flower as the origin of the reproductive structures is our starting point. So we start here with the flower. Within the ovary, we have the ovules. And within the ovule is the megasporangium. Meiosis gives rise to the megaspore. This time, unlike what we see in the gymnosperms, we do see the production of multiple haploid cells from the single megaspore that survives after meiosis. It divides 3 times and produces, sorry, 3 times. Yeah, I think it's 3 times. I'm trying to run the math in my head. Let me put it this way. It produces 6 haploid cells and a 7th cell that has 2 haploid nuclei in it. So one of the single haploid cells is going to become the egg cell. The remaining haploid cells are going to become supporting cells that will support the developing embryo. The cell that contains the 2 haploid cells, as haploid nuclei, will be the cell that will give rise to the endosperm. And it has a special role in fertilization. So following meiosis, we have the development of all these cells within the structure of the ovule, protected by the ovule wall. In the anther, you have the microsporangia containing the microsporocytes. Meiosis occurs within the sporocytes, giving rise to the microspore. That becomes encapsulated in a hydrophobic resistant coat that contains sporopollenin. The single haploid nucleus then divides to give rise to 2 haploid nuclei. One haploid nucleus is encapsulated and will become the sperm nucleus. The other nucleus is going to help generate the pollen tube once the pollen grain lands on the stigma and germinates if it is compatible with the flower that it lands on. And so one of the ways in maintaining the fidelity of pollination is simply by matching proteins on the surface of the pollen grain and the stigma, called pollen compatibility proteins. These must match in order for the pollen grain to germinate. So for wind-borne pollen, this is an important feature. Also for those plants that self-pollinate or that exclude self-pollination, matching pollen compatibility proteins or lack of matching proteins is a big deal in maintaining the fidelity of pollination. Now, remember, this is just pollination. This is not fertilization. So pollination is simply the arrival of the pollen on the stigma. Assuming that the compatibility proteins match, then the pollen grain germinates and the pollen tube starts to grow down the stigma towards the ovary. It penetrates through the wall of the ovary, grows around the edge of the ovule, and enters into the ovule via the micropyle, that gap in the integuments. Now here, the integuments typically consist of 2 layers of material rather than the single layer that we saw in gymnosperms, but in some angiosperms, you're going to find upwards of 3 layers of material. And they will dry out to form the seed coat, as we saw in the gymnosperm. So it's just a couple of extra layers, or an extra layer or layers of material as far as the integuments are concerned, but otherwise their roles are the same. So the pollen tube enters in through the micropyle and discharges the 2 nuclei. The sperm nucleus fertilizes the egg cell. The second nucleus, the tube nucleus, enters into that cell that contains the 2 haploid nuclei, creating a triploid cell. And that becomes the endosperm. So you can see that listed here. Okay. So this is the product of the second part of the double fertilization. So remember again, it's not a single nucleus that enters into the ovule, it's 2 nuclei. One nucleus fertilizes the egg, and so that creates the diploid condition that we expect for the zygote. The other nucleus enters into that cell that contains 2 haplonuclei. I guess you could say that it is functionally diploid, but the addition of that 3rd haploid nucleus now makes it triploid. And so that's going to be the endosperm tissue. So the endosperm tissue is derived from that triploid cell. So nutritive material is transferred from the parental plant into the endosperm and also to support the growing embryonic plant. When it reaches its mature form with its embryonic leaves, shoot, and root, water is extracted out of the endosperm and also out of the integuments, drying those out. The integuments turn into the seed coat and the little embryonic plant goes into stasis. Once the seed is released, if it lands in a suitable environment, which is typically presence of sufficient moisture, the moisture is taken in by a process called imbibition. So that's the proper name for the uptake of moisture that leads to the germination of the seed, imbibition. That allows the embryonic plant to start to grow again. It utilizes the resources in the endosperm to support its growth until the shoot emerges from the soil. The chlorophyll is then produced under the impact of the light, or as a consequence of the exposure to light. Remember, as I indicated earlier, those last couple of synthesis steps required light to trigger it. And so the leaves green up, photosynthesis begins, and the seedling then becomes self-supporting. and the seed and the endosperm then shrivel up, and the plant develops into the new sporophyte, or the seedling develops into the new sporophyte, and the cycle begins again. So that's the basic outline of the angiosperm life cycle. I'm just going to add a few more layers in here. Labels, sorry. It's not too terribly good. So that's separate. That's a separate step from fertilization. So the arrival, as I mentioned earlier, the arrival of the pollen on the top of the stigma is the pollination event. Fertilization is when the two haploid nuclei meet. So angiosperm evolution. This is, again, somewhat confusing. Darwin was very confused about the origin of angiosperms because the fossils that were available at his time suggested that angiosperms evolved very rapidly with very little fossilization. But that was because there were very few fossils to take a look at. We now recognize that, in fact, the evolution of angiosperms took place over a fairly extended period of time. There's not a huge amount of fossil evidence available, but it appears that the period during which angiosperms really emerged as a major clade in the plant kingdom was towards the mid to late Cretaceous. That's when we start to see a huge diversification of angiosperms. What we see in parallel with that is a diversification of insects. Now, given what we know about the relationship between insects and flowering plants as pollinators, an interesting question to ask is whether it was the diversification of insects that led to the diversification of angiosperms, or whether it was the diversification of angiosperms that led to the diversification of insects. The current thinking is that it was insects first, followed by angiosperms. And we'll see a little bit later just how that may have been triggered by looking at the basic two forms of flowering plant, of flowers in flowering plants. What we do know is that some of the very earliest fossils that show angiosperm features are two two fossils that come out of China. I have a picture of one of them called Archifructus sinensis. So if you look at the name, it means ancient fruit-bearing plant of China. That's what the fructus, the archi is ancient fructus fruit, sinensis is China. So this is a plant that we think represents an early flowering plant. And remember, you can't have fruit without flowers because a fruit is a mature ovary. So if you have flower-like structures, then you have the potential for fruit, hence the name. But what are being referred to as carpels and stamens don't have the protective feature of a flower. So there are no petals, there are no sepals to protect them. There is evidence of structures that look like floats. So it's been suggested that Archifructus was actually possibly a plant that grew in water. And so one of the questions that has been asked is, did flowering plants originate in water, or did they originate on dry land? Well, the evidence suggests now that in fact their origin lies in dry land, and that in fact, even though Archifructus has these structures that look like floats that may have held it upright in water, as an ancestor of flowering plants, this is not one. This is not a flowering plant or an angiosperm in the sense that we know angiosperms. But it does have carpels and it does have stamens, so it has features of flowering plants, but it is not considered to be an angiosperm because it doesn't have seeds. That being said, what do we know about the origin of angiosperms and seed-bearing angiosperms? So there is a group of extinct plants called the Bennettitales that have a structure that looks very flower-like. The thing that probably suggests that, again, this is not a true angiosperm is that it was not pollinated by insects as best as we can tell. It does not have a formal flower structure as best we can tell from the fossils, but it does have, again, angiosperm-like features. And so it is probably very closely related to modern angiosperms. And it was definitely woody, that we can tell. What further supports the notion that perhaps the ancestor of all modern angiosperms was actually woody and not herbaceous, and certainly did not grow in water, is that the very most basal representative of angiosperms, a flowering shrub called Amberella that is found only on the island of New Caledonia, in the scrubland found in New Caledonia, which is an island off the northern coast of Australia in the South Pacific. So it's north of New Guinea. It's actually French territory. And Amberella is a woody shrub. And it is also the most basal of all angiosperms, as we understand them. And so that suggests that, in fact, the ancestor of all angiosperms was probably some woody shrub-like plant that bore flowers. Now, what supports Amberella as being basal to all angiosperms is that some features of the way in which the reproductive life cycle of Amberella plays out is more gymnosperm-like than it is angiosperm-like. Likewise with the Bennettitales, they are still somewhat more gymnosperm-like than angiosperm-like. So that suggests that they are not true angiosperms. Amberella does have flowers, and it has true flowers with petals and stamens and sepals and everything. But again, the way in which parts of the reproductive portion of Amberella's life cycle plays out is more related to gymnosperms than it is to angiosperms. So that puts Amberella at the base of the angiosperm phylogenetic tree. The other basal flowering plants are the water lilies and the star anise and its relatives. So star anise and water lilies are also basal to angiosperms. Then you have the magnolids, which are the magnolias and their relatives, the black pepper and a couple of other plants that are included here amongst the mangolids, are also considered to be basal to the two major clades of the plant kingdom, the monocots and the eudicots. Now that split is relatively recent. We used to split the plant kingdom, or the angiosperms, into two clades, monocots and dicots. But when we start looking at the basal angiosperms, the Amberella, the water lilies, and star anise particularly, those were actually included amongst the dicots, and they have features that suggest dicot. And as such, if we're going to move them out of the dicots and place them basal to all angiosperms, then the original dicot clade is no longer valid, because you can't have part of the dicot clade sitting basal to all angiosperms and also have the rest of the dicot clade being separate from the monocots. That creates a paraphyletic clade, something that's not allowed under the phyllocode. So once the basal angiosperms were moved out and placed at the base of the phylogenetic tree, you had to rename the remaining dicots. So they used the term eudicot. And if you remember that the prefix U refers to true, then we now have eudicots as true dicots. Monocots is still valid as a clade. So we still consider this to be a valid clade under the current taxonomy. I guess I'll put an arrow down here just to... So here are the basal angiosperms. This is Amberella, and you can see that it does have stamens, it does have a carpel, it has petals, so it has a true flower, but the development of the eggs particularly looks very gymnosperm-like rather than, or more closely related to gymnosperms than angiosperms, so that places it right at the base. Then we have water lilies, next most basal, star anise as the next most basal, I guess. So these are the three basal angiosperms. Again, originally classified as dicots, now placed basal to the rest of the angiosperms. Then we have the magnolids, which are the magnolias, the laurels, that's the one I forgot, and the black peppers. These are still considered to be basal to the rest of the angiosperms, but they are more closely related to monocots and dicots, or eudicots, than they are to the true basal angiosperms. So this is a magnolia. If you've never seen a magnolia in bloom, if you're over on West Campus sometime, on the back side of West Campus, on the west side of West Campus, there are some magnolias that are planted alongside the road. So there's a row of about 3 or 4 of them, and they're usually in bloom early spring on into summer, and they have these very spectacular white flowers. So if you're there at the right time, you can actually see the magnolias in flower. I don't know of a number of any other places other than perhaps Lou Gardens or Mead Gardens that might have magnolias as part of their herbarium collection. So you could probably see them there, but if you don't want to pay to go see them, there are magnolias in the landscape on the west side of West Campus, in the little connecting road that leads out of the back of West Campus, that has the roundabout on it now. Over the lockdown period, they put a roundabout in the intersection on West Campus, on the back side of the campus. So if you're over on West Campus and you find the roundabout, then take the little side road that leads off campus, and there's about 4 or 5 magnolias along that little side road, and you can see them in flower if you're there at the right time. Then, of course, we have the monocots. These are the grasses and the orchids and the palm. So you could think of a palm as being a giant grass. These are all monocots, and they have typical monocot features. So the leaves are usually elongate with long and have parallel veination. That's very characteristic of a monocot. And so when you look at a grass leaf, whether it be wheat or corn or barley in this case, any of the grasses, they have the long leaves with the parallel veins. You see the same on the vein in the palms, and you see the same in the orchids. So that tells you that you're dealing with a monocot. Eudicots typically have a net-like pattern of veins, a broad leaf with a net-like pattern of veins. They have other features as well. So here are some typical eudicots. Here are the comparative features. So the name actually relates to the seed leaves. So a cotyledon, the cotyledon is the fancy name for a seed leaf. So a cotyledon is the embryonic leaf on the embryo. It's a seed leaf. So a monocotyledon has a single seed leaf. A dicotyledon has two seed leaves. Then you can see the vein pattern, parallel veins versus net. When you look inside the stems, vascular tissue in a stem is scattered in a monocot. It's in a ring in a dicot. The roots are fibrous on a monocot, whereas you have a single root with branches with what is known as a taproot system in a dicot. The pollen grain on a monocot has a single opening. Dicots have three openings. And then if you can look at the flower, the sporophyll arrangement in the flower, so the petals, the stamens, the sepals are arranged in multiples of 3 in a monocot and arranged in multiples of 4 or 5. Typically 5, but you'll see some flowers that have 4. Either way, you're dealing with a dicot under those circumstances. And you should be familiar with these differential characteristics. We also know that there is a strong relationship between angiosperms and animals. Animals have served as pollination vectors in the form of insects, birds, and mammals in some cases as well, have all served as vectors for pollination transfer, or we say pollination vectors. So here is a bee serving as a pollination vector. And you'll notice that in this case, it is pollinating a bilateral or bilaterally symmetrical flower. And the arrangement of the stamens almost guarantees that when a bee attempts to get to the nectaries at the base of the flower where the nectar comes from, that it will brush up against the stamens of a bilateral flower. And when it moves into another flower of the same species, it almost is inevitable that that pollen will rub up against the stigma at the top of the carpel of the flower because of its bilateral symmetry. And that leads to some very interesting observations about the rate of evolution and speciation in bilateral flowers versus radially symmetrical flowers. So bilaterally symmetrical flowers tend to be more routinely visited and pollinated by insects, whereas radially symmetrical flowers are more frequently pollinated by birds and mammals. So you can see a hummingbird here feeding from a flower. It has a long trumpet shape, which is the reason why hummingbirds have a elongated bill to reach the base of the flower. This is a case of co-evolution, where the flower and the beak of the hummingbird have evolved to accommodate each other. And some hummingbirds have extremely long beaks, and that is because they pollinate or they feed from flowers that have exaggerated trumpet shapes. You'll also notice that the vast majority of flowers that are visited by hummingbirds particularly, but by birds in general, have bright red or pink colors, because birds are able to see those colors, whereas insects see more lighter colors and can also see into the ultraviolet. And so they can actually detect patterns in flowers that are not visible under white light because they're only visible under ultraviolet. But of course, the sun provides the ultraviolet to allow them to see that. And then there are a whole range of flowers, particularly cacti, that are known as night flowering cacti. And of course, birds and insects are generally not out and about at night, but bats are. And so bats as mammals serve as pollinating vectors for these night flowering cacti. So let's talk for a moment about this relationship between bilateral symmetry and radial symmetry. Remember again, bilateral symmetry tends to favor insects and tends to attract more insects, and means that it is inevitable that the insect will pick up pollen because it's going to brush up against the stamen, the stamens and the anthers, and it will then brush up against the stigma on the end of the carpel when it visits a flower of the same species and transfer pollen. And because insects tend to be very true about which species of plants they pollinate, and because they get access to the nectaries and find it a little bit easier to find the nectaries and to also interact with bilaterally symmetrical flowers, that means that they are more likely to transfer pollen between individuals of the same species rather than spread the pollen around. That would not be the case if they made a living out of pollinating radially symmetrical flowers, because there the stamens are more widely dispersed, the carpels tend to be more widely dispersed. It's more difficult for them to get to the nectaries, and so they tend not to work the radially symmetrical flowers. That tends to be more the purview of the birds and the mammals that are involved in pollination. So what's the effect of insects primarily working bilaterally symmetrical flowers and being very I'm trying to think of an appropriate term here. Being very rigorous about working only certain species of flowers or working one species of flower at a time. It means that there is likely to be opportunities for greater diversification amongst bilaterally symmetrical flowers, because the insects tend to transfer the pollen within the same group of flowers that belong to the same species. If a mutation occurs within one group of flowers and the insects continue to work that one group of flowers, it creates an opportunity for reproductive isolation, because they're not going to visit flowers of a related species, because they maintain fidelity. That was the word I was looking for. They maintain fidelity to the flowers that they're working. And so because they continue to work the same flowers continuously, and they only transfer pollen within that group of flowers, any mutation that occurs is going to remain within that group of flowers, which you can see, as you think about it, creates opportunities for reproductive isolation. Whereas if they were working radially symmetrical flowers, and where they are more likely to relax that fidelity, that would allow mutations to be distributed more widely and collapse any reproductive isolation barriers. And this has been borne out by research. When you look at bilaterally symmetrical clades and compare those with radially symmetrical clades, there is a greater diversity of species within bilaterally symmetrical clades than there is within radially symmetrical clades. And that's because bilateral symmetry provides this greater opportunity for speciation by creating greater opportunities for reproductive isolation versus radial symmetry. So the actual shape of the flower actually contributes to speciation amongst the clades. So this is our final break before we cover the last set of slides relating to the plant kingdom. So let's take a look at this concept check question, and I will break again so that I can break the recording up into another section. So we'll see you on the other side of the break with the answer to this question. Excuse me. compare and contrast a pine cone and a flower in terms of structure and function. See you on the other side of the break.